Welcome back. In this lecture, we will discuss about proteins targeting. Protein targeting is also known as protein sorting. It is a biological mechanism by which proteins are transported to their appropriate destination in the cells or outside the cells. Proteins can be targeted to the inner spaces of organelles, different intracellular membranes, cell membranes, or to the exterior of the cell. This delivery process is carried out based on the information present on the proteins. The correct sorting or targeting is crucial for the cells, and errors can lead to diseases. Both prokaryotes and eukaryotes and eukaryotes newly synthesized proteins are delivered to specific subcellular location or exported from the cell to for the correct activities. In 1970, Gunter Blobel discovered that many proteins have signal sequences. These are short amino acid sequences at one end that function like a postal code for the target organelles. So the signal sequences or the targeting signals are the pieces of informations that enable the cellular transport machinery to correctly position a protein inside or outside the cell. So it helps the cellular transport machinery to correctly position the protein inside or outside the cell. This information is contained in the polypeptide chain or in the folded proteins. The targeting pathways. All proteins begins to be synthesized on the cytosolic ribosomes, and sorting or translation can occur by two pathways. First one is co-translation pathway. In this pathway, the transport of proteins occur during translation, which is not completed fully. So, and when the during translation, this type of pathway occur, which is known as translational pathway. Here, the synthesized protein is transferred to a signal recognition particles, which is also known as SRP receptors, which is present on the endoplasmic reticulum. There, then the nascent protein is inserted into a translocational complex and is sorted to different organelles. The next pathway is post-translational pathway. This is the pathway which occurs after the process of translation is completed. Most proteins are co-translationally translocated. Some are translated in cytosol and later transported to their destinations. And it occurs for proteins that go to the mitochondria, chloroplast, or peroxisome. So to the proteins which is sorted to these organelles are sorted by the post-translational pathways. So you can see here, this is the co-translational pathway, also known as secretory pathway. So this is the cytosol, this is the ribosomes, mRNA from which proteins are made. So this is the first one, this is the mRNA ribosome. So the proteins, this is the protein, this is the ER signal sequence which is attached to the proteins, and it it attaches here to endoplasmic reticulum, and then it is transported to various organelles. This is Golgi complex, this is plasma membrane, this is lysosome. So this is the secretory or co-translational pathway. This is the post-translational pathway. You can see here, this is the proteins. This is the cytosolic proteins, and these are the targeting sequence are present on this protein. So you can see here, this is first sorted to mitochondria, this is to inner membrane space, the matrix, next to chloroplast, inner membrane of chloroplast into the matrix, peroxisome, nucleus. So to these organelles, protein is sorted by a post-translational pathway. So these are the two types of pathways by which proteins are sorted to different cell parts or outside of the cell. Next is there are three fundamentally different ways by which proteins move from one compartment to other. So you can see here, this is the cytosol, and these are the three pathways. This is first one, this denoted by red is gated transport, the blue is transmembrane transport, the green is vesicular transport. So you can see this is the vesicular to endoplasmic reticulum, Golgi complex, lysosomes to exterior of the cell. This is the gated. The gated is used by nucleus, and this is the transmembrane mitochondria, plastid, peroxisome. So these are the three pathways by which proteins move from one compartment to another. The first one is the gated transport, which is used by the nucleus. Here the proteins move between cytosol and the nucleus through the nuclear pore complexes, which is present in nuclear envelope. The nuclear pore complex function as a selective gate. Gates actively transport specific macromolecules. So you can see here, these are the pores which is present in the nucleus, and these act as a gates and allow the transport of specific macromolecules. The next one is transmembrane transport. This occur in mitochondria, peroxisome, chloroplast. So in this, the transmembrane protein translocators are directly transport specific proteins across membrane from cytosol into the space. So here, the transmembrane proteins translocator, which is present in the membranes, and it translocates the proteins from cytosol into the membrane, across the membrane of mitochondria, peroxisome, and chloroplast. Proteins molecules usually must unfold to go through translocator. So in this process, the protein first unfold to go to these translocators. The last one is vesicular transport. It is used by endoplasmic reticulum and Golgi apparatus. Here the membrane enclosed small spherical transport vesicle or larger irregularly shaped organelle fragments move proteins from one compartment into another. So in this, the membrane enclosed small spherical transport or largely irregular shaped organelles fragments move proteins from one compartment to another. So you can see here, this is the endoplasmic reticulum. This is the donor compartment. You can see this is budding vesicle with contents selected for transport. This one. So it budded transport vesicle in the cytoplasm. This is the targeted compartment. So it fused with it and it released the targeted protein. So this is the vesicular transport system used by the endoplasmic reticulum and Golgi apparatus. So this is all about protein targeting. Thank you for watching.